Okay, so we're going to continue with the eye and vision. And first thing we'll talk about are what we call accessory structures to the eye. These are not directly involved in vision, but aid in that sense by protecting the organs involved in seeing or other protective functions for the eye itself. So some of the accessory structures, we have eyebrows. Everybody knows eyebrows, they shade the eye. They also prevent respiration from going into the eyeball. You didn't know that. That's the reason that we have. So every single time you don't have a big sleepy eyebrow, you're actually pulling and plucking, you're actually doing yourself a disservice. You're going to get sweat in the eye. Eyelids serve a purpose of protecting the surface of the anterior portion of the eyeball. We'll talk a little more about it in a little bit. Eyelids and then eyelashes as well. So eyelashes are connected to our response or reflex to be able to close our eye very, very quickly in response to something coming close to our eye. So we have a natural reflex to close our eyeball to protect us. And we don't want to get a stick or something else in the eyeball that can actually scratch or damage the cornea. The conjunctiva, again, whatever you want to call it, conjunctivitis. The guy that I learned this from was Belgian. He always pronounced it conjunctiva, but conjunctiva is really the correct pronunciation as I know it. Conjunctivitis is what you always hear of. So it's hard to see this. It's hard to understand this idea. But right here, there's actually a membrane that's actually right up here of the eyelids, kind of curves around and covers just the surface of the eye. So this is not the cornea, it's outside of it. Okay, it's superficial to the cornea, and it's called the conjunctiva or conjunctiva. And it's this little membrane right here that protects the surface of the eye. This membrane can become inflamed by bacteria or viruses or other infectious agents, which is where you get the term conjunctivitis. So if you get an infection of Haemophilus influenza or some other bacteria, they get in the eye, we have pink eye. So pink eye is just bacterial conjunctivitis. That's what that is. You can have viral and other things as well. But that's what that actually is. So it's hard to see it because it's very, very clear. But again, it's just right here on the actual surface of the eye. Now, when people mostly talk about the eye, the first thing they talk about is the cornea. Technically, that's not the most superficial layer. Technically, it's the conjunctiva. But people sometimes forget that. Just thought I'd point that out to you guys. The cornea is what we'll talk about here, and that's actually what serves as the glassy, I guess, outside portion of the eye. We'll come back to that in more detail in just a little bit. Okay, as far as here's our concerns. We have an apparatus we call the lacrimal apparatus. Lacrimal secretions are called tears. Lacrimal secretions are actually produced by a gland called the lacrimal gland right here. And the secretions are used to coat the surface of the eye. So right on the conjunctiva. This serves to lubricate the eye so we can actually close our eyelids. Prevents it from drying out, which is not a good thing when it comes to the surface of our eye. And actually also has antimicrobial agents in it. So there's things like lysozymes, which you haven't studied unless you take it in microbiology, that actually break down bacteria. The overall result is the surface of the eye is not sterile, but it's cleaner than most of the other surfaces of the body. Because it's constantly being lubricated and mechanically being washed and has enzymatic action against bacteria that basically keeps that area of the eye cleaner than most. That surface of the body cleaner than most. So lacrimal secretions then get drawn into and go into the nasopharynx, which is why when you cry, you always get a stuffy nose.
So why is it if you cry too much, your eyes get puffy?
I don't know that. I don't know. It could just be the buildup of the fluid, edema. I don't know. I've never been asked that. Yeah, good. Interesting. I can tell you this. You build up those secretions at night and they dry out. and you end up getting nice little crusties in the corner of your eye. They actually call that Sandman's eye sand. If you ever wonder what that's called. Really, that's what the technical term is, Sandman's eye sand. You call it, you know, sleepy seeds, goobers, whatever you want to call it. We saw how 3 or 4 different cranial nerves are involved in moving these muscles. We really didn't talk about these muscles specifically, and you don't have to know them by name. But big surprise, not surprising I should say, you know, we have multiple different muscles that are going to move the eyeball. These muscles are very, very rapid muscles. We can move our eyeball very, very fast. You know, left or right, up or down. We can, you know, move it in circles, etc., etc. We can move all these around. Again, this is just simply moving the eyeball, not vision. It's not involved in vision. Move the eyeball to direct our main focus on different parts of our environment. So the frontal eye field is what's involved in this. If you go back to the brain, the frontal eye field moves the actual eyeball itself, but it's not directly involved in vision. This is a picture that will pop up on your exam. Okay? Maybe the only one of the eyeball that'll pop up on your exam. Maybe. And this really shows you a lot of the things we're going to be talking about, if not all the things we're going to be talking about with respect to the eyeball. So we look at the eyeball, and it's fairly spherical, round in shape. Okay? And we have a nice tail that comes off the end of it, which we're going to call the optic nerve. So we're going to have a bunch of cells that are going to be in this eyeball. They're going to pick up photons of light, and they're going to detect that stimuli and send their nerve impulses, send the action potentials all down here to a nerve we call the optic nerve. And we'll see more about this detail in just a few minutes. So when you get your eyeball today, you will get an eyeball later on, you're going to get an eye that looks like this. It's still going to have, hopefully, some of this will attach so you can actually see the optic nerve. Now, most people are going to end up cutting it apart and realize it should have it attached. And I'll try to get you one that has some attached. So when we talk about the layers that make up the eyeball, if you look at this picture, we have a couple of layers. We have what's called the fibrous layer. The fibrous layer is, well, as you would imagine, fibrous, or it's protective. It's the strong, tough layers that make up the eyeball. So think about the eyeball. When you actually get the eyeball today, it's going to be harder than you think. You think it's going to be kind of squishy. It's not going to be so squishy. It's going to be rather hard. And that hardness of the eyeball is needed because, go back to those muscles. These are going to be pulling and twisting that eyeball. That eyeball has to be fairly rigid. And so you have to have some sort of fibers or fibrous structure on the outside of the eyeball to handle the forces of turning that eyeball in all kinds of directions. So we have fibrous layers. We're going to call that the sclera and the cornea. So the sclera is the whites of the eye. Okay, the whites of the eye. And you're going to see this later on, but if you look at the eyeball, the entire eyeball will have a white layer except for where the cornea is. So I'm sorry I don't have white, I can't draw white, I'm doing blue. But this is the sclera in white. Okay? All around the whites of the eyeball is called the sclera. And then the cornea is this convex meaning outward, clear, not white, but clear kind of window to the inside of the eye. So when people say, you know, the white of the eye, you ever heard that? Don't shoot until you see the whites of the eye. World 1812 or I can't remember which one it was. One of those we learned way back. Right? The whites of the eye. That's called the sclera. The cornea, as I said, is a transparent anterior bulge. We'll talk more about this in a little bit. The cornea is the window that allows light light energy to go into the eye. It's extremely vulnerable to damage though. But it's also extremely repairable. It's amazing how fast you can repair a scratched or cut cornea. Anybody ever scratched their cornea? For that 24-hour period, you want to kill somebody because it hurts so badly. But after 24 hours, you're practically healed. It's amazing how fast it heals versus the skin. The cornea is a very impressive structure of the eye. And there's one mention here. Other things that are just interesting, but nothing I need to go into. The vascular layer. Vascular, that means blood vessels, right? So we're going to have some vascular layers. We're going to have what's called the choroid. We're going to have what's called the ciliary body. We're going to have what's called the iris. There's going to be what's called the vascular layer. And so if you look, and I'll show you maybe some better picture. That's probably the best picture I'm going to have. So here's your white sclera right here, the outside of the eye. And you can see right here how it continues wrapping around the optic nerve. So there's the sclera. It goes all the way around and stops right here because this is where the cornea is. And then here we have the sclera again. Okay? Just inside that, just deep to that, we have this, the brown, called the choroid region. Highly vascularized. It's dark, okay? But it's also full of blood vessels. Why is it dark? Well, let me explain something. Go to a movie theater. Why are the walls painted dark?
So they don't reflect light.
Don't reflect light, right? Here we're trying to get a very precise picture on the back of our retina, which we'll talk about in just a few minutes. If light bounces all over the room, we're not going to have a very distinct, crisp picture, are we? So we want to minimize the amount of light that's bouncing around so the walls are painted black. And you're going to see that when you cut open the eye today. You're going to see that nice black layer, and that's the choroid region. So it's a highly dark brown membrane, highly vascularized. And that's actually what's going to feed the rest of the eye. The iris is right here, and the iris is what you would call the color of the eye. Someone has green eyes, they have blue eyes, they have black eyes, brown eyes, whatever the case may be. That's the iris. And really what the iris is a muscle. It's a muscle. And they're trying to show that here. You can kind of see how it's cut, so you can see that muscle. And it's held by the ciliary bodies. Well, kind of. The ciliary bodies hold the lens too, but that's what's right here. So let's talk about the iris. The iris is that has pigment. Pigment in it. Pigment is, well, color, dye, right? Would it surprise you that the pigment inside your eyes is the same pigment inside my eyes, no matter what color eyes you have? I don't care if you have green, blue, purple, pink, whatever eye color you have. It's the same dye, the same pigment. The only difference between someone who has blue eyes, green eyes, black eyes, and brown eyes is how much pigment is there and where it's positioned within the iris, front to back. Don't believe me, every baby is born with blue eyes. Every one. Blue is the natural color of the iris. After a while, meaning a few months after you're born, the baby starts to produce pigment if it has a gene for it. So the eyes go from blue to green to maybe eventually hazel, right? But if you have blue eyes, you just don't have the pigment. You don't have the gene for it. So color, or brown eyes, go back to the genetic idea of it, is what you call a dominant gene, right? Remember the idea of dominance? So it's a dominant gene. So if your dad has blue eyes and your mom has blue eyes, I guarantee you have blue eyes. There's no way you couldn't. But your mom and your dad could both have brown eyes, and you could end up having blue eyes. Just depends on how Mendelian genetics works out.
Gray eyes.
Gray eyes, well that's different.
Yeah.
Yeah.
With two brown-eyed parents.
Wow, yeah. It's weird. And people can have different color eyes too. It's rare. But you can have it. It happens.
So my sister, she's got really light blue eyes, and mine are darker. So does that mean I have some pigment?
Yes. And you can have all kinds of gradations of it too. I mean, you know, my wife's eyes are blue some days and green other days. It's just, she's got a little bit of pigment, it's just how it's positioned within the iris. So the ciliary body is right here, just so we won't talk really much about it, right here. And it kind of holds this together and really specifically holds the lens in place. And we'll talk about the lens in more detail in just a few minutes. The inner portion of the eye, we call the retina. The inner portion. And this is really where vision takes place. And so here, what they're showing you here is in yellow. So all this in yellow right here, they're calling the retina. Okay? Now we're going to see different portions of the retina, and we'll talk more about that in just a few minutes. Okay? Yes, we'll come back to this idea. I'm going to point this out real quick. Macula lutea. Now don't confuse that with that. Just a little bit of a hint. The macula is involved in equilibrium. The macula lutea is involved in vision. People sometimes get confused. I won't ask a question it has both in there, but I don't want you to get confused when you see that versus that. It's spelled exactly the same. Just got that macula lutea. So we're going to talk more about this area in just a few minutes. But what I want you to think about is, okay, here's the retina. This is the portion of the eye that's going to detect light. Black, whites, grays, blues, purples, all different colors, right? And so you can imagine that we're going to have different receptors that are designed to pick up different colors. And specifically, we have a certain amount of cells that are designed to pick up our blacks and whites, or our grays, okay? We call those cones, or rods, sorry. And we're going to have our cones, we'll get to in just a little bit, they'll pick up our color, okay? So we're going to have different proportions of rods and cones kind of together. Right here is where we have almost all colors. All of our cones are right here. And specifically, this area right here, we call the fovea centralis, is all cones. Again, we'll come back to this. I just wanted to point this out because it's on this picture. I got a better picture for it in just a few minutes. But before I move on, what I want you to think about is here's your eyeball, right? Imagine this is a ray of light, a laser beam. We'll make it real simple. A simple ray of light is going to come right here. The very first thing it's going to hit, even though it's not showing you this on the picture, is the conjunctiva. Then it's going to go to the cornea, pass through the cornea, pass through the pupil, which is the hole in the iris, then hit the lens. Then it's going to be bent. We'll talk about how it'll be bent later on. But let's just say for now we're going to go straight through, okay? Right to the very, very back of the eye, right there. So if we're looking at something dead on, straight in front of us with our eyeball straight at it, that photon of light will go straight through the back of our eye. And the highest visual acuity that you have is straight in front of you, isn't it? That's that area right there that you're hitting. Okay, we'll come back to that. The fovea centralis, and then surrounding that's the macula lutea. Make sense so far? So we're talking about the light coming in this way. So it's going to be hitting the back side of the eyeball right there. Here we are again. Now, I wish this was oriented this way, but it's not. So the light is going to come in this way. This we're seeing right here is the retina, blown up, really, really big. And there's going to be several different types of cells here. You need to know all of them. Okay? Unlike smell, I want you to know these. Vision is important, right? For this. The thing that confuses people, the thing that people don't always understand is the direction of light. So it's going to come through here. It's going to pass through several cells on its way to these cells right here. So it's going to pass through the ganglion cells. It's going to go right past the bipolar cells and go right to and be picked up by the photoreceptors we call the rods and the cones. So this here is the back of the eye. Okay? So right after this layer, we would have the choroid and then the sclera. Okay? So again, the direction light's going this way. These two types of cells we call the photoreceptors. We have two types. We have the rods and we have the cones. They are named rods and cones because this portion of the cell looks like a cone and this portion of the cell looks like a rod. Okay? That's why they're named that. Not very ingenious people. Oh, look at the cone, looks like a rod. Okay? The cone is involved in picking up colors. And the rod is involved in picking up black and white. Or grays. Really not variations between black and white. We'll talk more about this as we go on. This is going to give us very low visual acuity. What do I mean by that? Well, put yourself in a dark room with just a little bit of light. You're not really seeing colors, right? You're seeing shadows. You're seeing distinct patterns as far as you can tell where the table is, where the desk is, or you're wandering around a dark room, you can see just a little bit, right? Low visual acuity. You're not really seeing color. All you're really seeing is grays and whites, right? And it's not real sharp. Make sense? It's also what you predominantly see in your peripheral vision, although you do see some colors. Okay? What you're mostly seeing is the basic outlines as defined by the rods. So low visual acuity. These right here are your cones. They are involved in high visual acuity. This is your HDTV right here. Okay, high visual acuity or high visual acuity. These are involved in color. Now this picture is good and bad, but I'll show you the part that's good. Okay, and it's this. What I want to point out to you here is this versus this. Okay? Now, imagine a photon of light comes through. Okay? This photon of light is going to come through and hit the cell right here. And it's going to respond. We're going to talk more about how they respond. There's pigments that respond to light energy, and they're designed to pick up a certain frequency, a certain wavelength. Okay, so blue, 520, blue, or red, different wavelength lengths. We'll go more into that. So each one of these is designed to pick up a particular wavelength of light. So this guy right here will pick up a blue. And this one right here will pick up a slightly bluish green. A little different maybe. My point here is that look where these guys transmit. So this one responds. This one responds. This one sends information this way, which synapses to this bipolar cell, this ganglion cell, and then this ganglion cell will send a single fiber over to the brain. This one here responds, synapses with a different bipolar cell, a different ganglion cell, and goes to the brain. So the brain can tell the difference between this one firing and this one firing. Right? It's just like we talked about before. I can feel two pricks in my hand, because we're responding differently. But look at this guy. This guy receives a photon of light, or this one receives a photon of light. And in both cases, one bipolar cell, that's not really showing you here, one ganglion cell. And what ends up happening is, well, this is why your vision with black and white is not high visual acuity. This is why you don't have HD black and white vision. Because you have very few bipolar cells dedicated to the rods. Make sense? Kind of?
Okay, but so that makes it look like the bipolar cells and the ganglion cells are in the middle, are floating around in the middle of the eye.
They are. They're not in the middle of the eye. This is, all this right here that you're seeing, from here to there, all of that is right here in this yellow. That's a yellow layer. Just in the back right here. See that little layer right there? All of this right here is liquid, fluid, called a vitreous humor. We'll get to that. Okay? All we're talking about here is this little cell layer right there. And it's really thin, as you're going to see. You're going to pull it off the back of the eye. Really, really thin. And all of that we just talked about is that little thin layer right there. So you have that single cell layer at the back of the eye, the back of the retina, that are the responders, the photoreceptors, as we'll call them. So name the two photoreceptors, rods and cones, right? Then you have the bipolar cells, then the ganglion cells. And it's actually the ganglion cells, the axons of those ganglion cells, that will all come together and form the optic nerve. That's what that is. So if you go back to the eye, big picture, now you can imagine you have those ganglion cells all the way along the entire surface of the retina, picking up information from rods and cones, and every one of those axons is going to go down here and form what we call the optic nerve. Okay? So the interpretation of this will be, okay, are we seeing color? Are we seeing black and white? And where in the eyeball is it coming from? That's the interpretation we're going to get. Something of interest, not something I'll test you on, but something that's kind of cool with the eye. Here's the retina. We can see anything. If the light gets bent up here, 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 or here, or here, anywhere along the surface of the retina, we can see. But right here, this is where we have all the axons going into the eye. There's no photoreceptor right here. We have a blind spot. Technically, we have a blind spot in your eye. So it's not what you look at directly, it's just a little bit below that. Okay? Kind of toward the center of the medial side of the body. That is a blind spot. So technically, you have a blind spot in each eye. But the brain fills it in. It uses information from what's around it to fill it in so you can't really detect that blind spot. Now, you can't do this in the classroom because I've tried and people can't really do it. But there's a test. You can do something like this at home, where you can close the eye, stare at the X, and the red dot will disappear because it's in your blind spot. Okay? There's a lot of little tests like that you can do. But all those little tests where you look at something for a long time and things disappear, that's because of your blind spot. It's just you trick your brain into forgetting it's there. So here's another way of looking at it. So again, look at the back of the eyeball. And there's your fovea centralis. We'll talk more about that in your macula lutea. And there's your blind spot right there. So again, two photoreceptors. We'll talk a lot about rods and cones. So two photoreceptors. We have our rods. Well, let's talk more about them. What are they? Well, there's a lot more rods than there are cones. A lot more rods than there are cones. They respond to dim light. In other words, they are designed to respond, to activate, to send an action potential in response to dim light. So it could be very, very dark outside. There could be one little faint bit of light, and that's how you're going to see. And it's the rods that are going to respond to that. Your cones don't activate. You need more light to turn them on. Okay? It's the rods that are activating by dim light. They're primarily used for peripheral vision. So if you go back over here, what you're going to see, go back to the main picture. If you were to count the number of cells, let's say I were to take a picture and grab 100 cells right here, 100 photoreceptors. This area right here, in this area, toward the back of the eyeball, is going to be, the majority of them are going to be cones, color. Very few rods. But as you go further and further away from the center, you're going to have more and more rods. Less and less cones. So over here, you're going to have all rods. And right here in the center, in what they call the fovea centralis, is 100% cones. And right around that general area we call the macula lutea is the majority of it is cones. But again, the further and further away you get from that, the more rods you have, the less cones you have. But again, they do not provide sharp vision, the rods don't. And they don't provide color vision. They're only involved in black and white or grays, best way to think about that, and not very high visual acuity. To really see high visual acuity, you need cones. Cones respond to bright light, and they give us our colors. So we have three types of cones that respond to three different colors, but we can blend those three colors to make any color in the visual spectrum. Okay?